Everybody. All right, welcome to chapter 17, where we are talking about the citric acid cycle. For some reason, I put down sections 1, 2, and 3, but section 4 is also a good section to read that has to do with the regulation of the citric acid cycle. So in this lecture, I'm probably not going to go as in-depth as the chapter goes on some of the enzyme mechanisms. That's for a more advanced course. However, this will kind of give you a good outline to work from as you prepare for class. Remember, so this lecture is going to be one total video, although the class itself will be broken up into 2 class meetings as we work on some of our group activities and discussions for this chapter. Okay? So here are your learning objectives. All right, so feel free to pause here, read these over, and make sure we understand what we need to keep in the back of our mind as we go through the rest of this lecture. Okay, let's continue on. Okay, so the citric acid cycle is our next step in the metabolism of glucose. So we can start from glycogen, get glucose, go through glycolysis to get pyruvate. Pyruvate is then converted to acetyl-CoA, giving off a molecule of CO2, and then it gets shuttled through the citric acid cycle to further produce more CO2, as well as some NADH, ATP, and FADH2. Okay, so these molecules of FADH2 and NADH are then sent to the electron transport chain to generate more ATP by transferring our electrons through oxidative reduction reactions. All right, so the citric acid cycle occurs in the mitochondria for the most part. So if pyruvate was developed in the cytosol, it has to be transported into the mitochondria using some transport proteins, and we'll talk about that in just a second. So as reduced carbon atoms of metabolic fuels, such as glucose being converted into pyruvate, are oxidized to CO2, electrons are transferred to electron carriers, such as FADH2 and NADH, and then eventually to oxygen in aerobic organisms, so oxygen being our final electron acceptor. The citric acid cycle can recover energy from several metabolic fuels, not just glucose. As you see, amino acids and fatty acids can also be broken down to generate acetyl-CoA, which can then be shuttled through the citric acid cycle. There are 8 reactions total that are used to oxidize the acetyl groups of acetyl-CoA to 2 molecules of CO2. The energy released in that oxidation is conserved in the reduced compounds of NADH and FADH2. This cycle was discovered in 1937 by Hans Krebs, so it's often called the Krebs cycle. And then we also can call it the TCA, which stands for the tricarboxylic acid cycle. Often abbreviated as TCA, which you'll see me abbreviate as TCA often. So overall, 1 acetyl-CoA that comes through the citric acid cycle, plus 3 NAD+, an FAD, a GDP, an inorganic phosphate, and 2 molecules of water are converted into 2 molecules of CO2, 3 molecules of NADH, 1 molecule of FADH2, a molecule of CoASH, GTP, which can also be either ATP, and 2 acidic protons. Okay, so there we see our NADHs, FADH2, that GTP, and our 2 molecules of CO2. So the TCA, or the Krebs cycle, oxidizes acetyl groups from many sources, not just pyruvate, so we already talked about that. The oxalo acetate that is consumed in the first step to make citrate is regenerated in the last step. So that's why it's cyclic. All the enzymes for the TCA are in the mitochondria. The 2 carbons of the CO2 molecules brought in the acetyl group on acetyl-CoA are not actually the molecules that are extracted out as CO2. These are lost in sequential rounds, and we'll sort of see that as we follow the reaction pathways. The TCA intermediates are precursors for the biosynthesis of other compounds. So we'll see that alpha-ketoglutarate, isocitrate, malate, succinate can all be used in other metabolic pathways. And then a total of 4 electron pairs are transferred from acetyl-CoA to make NADH or FADH2. After the electron transport chain, these 4 electrons can generate a total of 10 ATP when transferred to oxygen in the electron transport chain. So, how do we get acetyl-CoA from pyruvate? So first, pyruvate is transported into the mitochondria with a transport protein. We also co-transport an acidic proton in with us. Acetyl-CoA is formed from pyruvate through an oxidating decarboxylation reaction using a multi-enzyme complex known as the pyruvate dehydrogenase. So this complex is actually composed of 3 enzymes, pyruvate dehydrogenase, which is also called E1, this molecule, which is called E2, as well as E3. The pyruvate dehydrogenase complex is regulated through either direct product inhibition, okay, so acetyl-CoA will inhibit it, covalent modification, so the E1 protein requires phosphorylation, which this is triggered through glucagon release. It is also activated by NAD+, CoASH, or AMP, and then inhibited by ATP, acetyl-CoA, again, remember its product, and NADH. So in order to do this conversion, the first step, E1, uses a cofactor, TPP, here, to remove a CO2 group from pyruvate with the formation of a hydroxyethyl TPP intermediate that we see here. This intermediate is then transferred to E2, where it attacks the lipoamide disulfide, and TPP is eliminated. The carboanion intermediate is then oxidized to an acetyl group as the lipoamide disulfide is reduced. E2 then catalyzes a transesterification reaction in which the acetyl group is transferred to give us an acetyl-CoA. So technically, this is sort of where our story ends in the production of acetyl-CoA. However, we need to regenerate the lipoamide group of E2. This is done through the oxidation of E2 by E3, the reduction of E2 by E3, so that we can regenerate the lipoamide and the enzyme can continue. Then E3 needs to be reoxidized utilizing a NAD+. So here we actually generate another NADH. However, this NADH generation is not considered part of the citric acid cycle because we don't start the citric acid cycle until we have our acetyl-CoA. So this is just another additional bonus of NADH. So if we look, pyruvate dehydrogenase, if you may have noticed, has a number of different cofactors. Thiamine pyrophosphate, TPP, lipoic acid, coenzyme A is a cofactor for E2, FAD is a cofactor to E3, and then the NAD+ is a cofactor of E3 as well. So it's a very, very busy enzyme complex. And if you look in your textbook, it talks about the importance of utilizing enzyme complexes such as pyruvate dehydrogenase in section 2A. So that's a very good thing to read about, the advantages of why you would use multi-enzyme complexes as opposed to having all of these enzymes separate and on their own. All right. So now that we have acetyl-CoA, we can begin the citric acid cycle. So step 1, we introduce 2 carbons to acetyl-CoA through a condensation of acetyl-CoA with oxaloacetate to form citrate. So we lose that CoA in the reduced form, giving us citrate. So the hydrolysis of the intermediate in this step releases 31.5 kilojoules per mole of energy, which gives us the driving force of this reaction. In step 2, citrate is isomerized from a secondary alcohol to a more reactive, from a tertiary alcohol to a more reactive secondary alcohol. This reaction is reversible. However, we do have a slightly inorganic reaction. However, this is pulled forward because isocitrate is rapidly depleted in the following reaction steps of the citric acid cycle. Alright, so our next step, step 3, is isocitrate is then oxidized to form our first citric acid cycle molecule of NADH, releasing a CO2 molecule. This oxidative decarboxylation is catalyzed by isocitrate dehydrogenase. It requires a cofactor of either magnesium or manganese. Right. This reaction results in the formation of an enol intermediate that rearranges to form an alpha-keto acid. Okay, so alpha-ketoglutarate. Again, this is where we produce one of our first NADHs and CO2s. In step 4, alpha-ketoglutarate is oxidized to form a second molecule of NADH and CO2. The conversion of alpha-ketoglutarate to succinyl-CoA is catalyzed by the enzyme alpha-ketoglutarate dehydrogenase complex, much sort of like the pyruvate dehydrogenase complex. This reaction is also highly exergonic with a negative 33.5 kilojoules per mole. You'll see that this enzyme is actually inhibited by succinyl-CoA, NADH, ATP, and GTP. So this is sort of one of our big control points in the citric acid cycle. Step 5, we have cleavage of the succinyl-CoA coupled with substrate level phosphorylation. So cleavage of the high energy thiester bond of succinyl-CoA to form succinate is reversible and catalyzed by succinyl-CoA synthetase. The direction of the reaction depends on the relative concentrations of the nucleoside diphosphates and the nucleoside triphosphates. So if we have plenty ATP or GTP, the reaction will not occur. But if we have a high level of ADP or GTP, we'll see that the reaction will proceed in the forward direction. In step 6, succinate is oxidized to form fumarate and FADH2. This enzyme reaction is catalyzed by succinate dehydrogenase. This enzyme is found tightly bound to the inner mitochondrial matrix, and we'll see why that's important when we talk about the electron transport chain. Here, we're sort of near zero, slightly exergonic. It's a negative 5.6 kilojoules per mole. This enzyme is activated by succinate or ADP and inorganic phosphate, while it is inhibited by a downstream product, oxaloacetate, as well as malonate. Malonate is a byproduct that accumulation of malonate redirects the energy of fatty acid synthesis. So if we have plenty of energy, we have plenty of ATP, malonate will build up and say, hey, instead of making more ATP, let's start building us some fatty acids. Okay, in our next step, fumarate is hydrated, forming L-malate in a reversible stereospecific hydration reaction that is catalyzed by the enzyme fumarase. In step 8, our final step, malate is oxidized to form oxaloacetate. So now we're back at the top of our citric acid cycle, producing our third molecule of NADH. This is catalyzed by malate dehydrogenase. And it is a very endergonic reaction, so it's plus 29 kilojoules per mole. So that's a delta G standard. Okay. This reaction, however, does proceed forward and becomes closer to a delta G of 0 because oxoalylate is rapidly removed in the next round of the cycle in another round of the citric acid cycle. Okay. So here is sort of a nice overview of the whole cycle. You can see pyruvate coming in, getting condensated with oxoalylate to make citrate, isocitrate, alpha-ketoglutarate. Anyway, I thought this would just be a nice figure for you guys to have. You can print out and sort of go over and analyze everything in here. So the citric acid cycle metabolites can be used for biosynthesis as well. So as you can see, this figure here is from your textbook. You can see that citrate can be used to make cholesterol and fatty acids. Alpha-ketoglutarate is used to make a number of different amino acids. Succinyl-CoA can be used to make porphyrins. Remember, this is found in your heme. A couple other amino acids, as well as odd-chain fatty acids, so fatty acids that have maybe 31, 11, 15, okay? Fumarate can be used to make some other amino acids, aspartic acid, phenylalanine, tyrosine. Malate can be used to make glucose. Oxoalate can be used to make also amino acids. This figure is from another textbook, and I always like to give you a couple different perspectives. Here, we can see oxoalacetate can be used to make aspartate, as well as can be shuttled back in for gluconeogenesis to synthesize glucose, and can be used for pyrimidine synthesis. Citrate, again, can be converted back into oxoalacetate. The acetyl-CoA can also be used. Remember, we saw that in fructose development, but can be used for fatty acid and cholesterol synthesis. Alpha-ketoglutarate can be used to make glutamate as well as purines in nucleic acids. And then again, succinyl-CoA, certain fatty acids and amino acids can be broken down to form succinyl-CoA. And then succinyl-CoA can be used to make hemes and chlorophyll, which is actually a type of heme. Here is sort of an overlook of our products of the citric acid cycle. So remember, we're going to form 3 molecules of NADH, 1 molecule of FADH2, a molecule of GTP, which is also kind of synonymous. We can get freely phosphorylation of ADP to make ATP, and then our molecules of CO2. So if we look at the metabolism of glucose starting from the top, if we go through glycolysis, remember we make 2 NADHs in glycolysis. So each NADH can make about 2.5 molecules of ATP per NADH. So for 2 of those, we're going to get 5 ATP. And then we also generate 5 ATP directly. So NADH through the electron transport chain makes 2.5 ATP. So for every 1 NADH, we eventually will get about 2.5 molecules of ATP. Pyruvate, remember when we convert pyruvate to acetyl-CoA, we're going to get NADH. So, in glucose we get 2 pyruvates. So overall that gives us 2 NADHs, so another 5 ATP. Now that we have 2 acetyl-CoAs for every glucose molecule, we're going to go through the citric acid cycle twice. That will give us a total of 6 NADHs for 15 ATP on average. 2 FADHs give us an average of 3 ATP, and then 2 GTP, which also translates to 2 ATP. So total, that gives us 5, 10, 25, 26, 27, 28, 29, 30, 31. So about 32 ATP for every molecule of glucose if we start with just straight glucose. All right? So think about what our total ATP would be if we started, say, with acetyl-CoA or we started with just one pyruvate. All right. Now, we kind of mentioned some of the regulation factors for the citric acid cycle when we were talking about each step individually. But here's just sort of a nice diagram of some of the enzymes that are stop points, indicated by these red dots, as well as activation points. Okay, so if you remember, pyruvate dehydrogenase has to first be phosphorylated before it becomes activated. And if we dephosphorylate it, so insulin, if you read in your chapter, actually causes the dephosphorylation of pyruvate dehydrogenase, so that will stop dehydrogenase. But also, pyruvate dehydrogenase can be inactivated by acetyl-CoA, ATP, and NADH. Granted, this isn't really considered part of the citric acid cycle yet, but we're going to talk about it anyway. Also, pyruvate can be converted to oxoacetate through pyruvate carboxylase, which is stimulated by acetyl-CoA. Okay. But we are going to burn a molecule of ATP doing it that way. Okay. In the citric acid cycle, we see that we actually have 3 enzymes that are big points of control, and surprise, surprise, they happen to be the enzymes that have a negative delta G in the cell. All right, so your negative delta G standards are here. So this is if everything is thermodynamically under standard conditions. We see that most of the reactions are either very negative, near zero. However, the malate dehydrogenase we see is actually fairly very positive. However, under conditions in the cell and due to rapid removal of these enzymes products, we can take these very thermodynamically unfavorable reactions and make them near zero and likely to occur. However, we do see that in reactions 1, 3, and 4, where these reactions are very thermodynamically negative and very thermodynamically favorable, we see that these also correlate to points of enzymatic control. All right, so citrate synthase is inhibited by NADH and ATP. So, you know, why go through the citric acid cycle if we already have plenty of ATP and NADH? As you see here. And it is also inhibited by citrate and succinyl-CoA. Isocitrate is inhibited by NADH and ATP as well. However, it is also stimulated by NAD+ and ADP. Alpha-ketoglutarate dehydrogenase is inhibited by succinyl-CoA, which is its product, as well as NADH. Okay, so that's all I wanted to go over in this lecture. We'll talk about this in more detail in class as we go through those POGIL activities. And if you have any questions, feel free to Facebook post or email me or come see me. All right, hope you guys have a great night, and I will see you in class next time.